Hi, I'm Timmy the Turtle, and next to me is my sister Tina. Our bodies were cut out of phenolphthalein gel. You're probably wondering why we're sitting in a petri dish. The answer is science. More specifically, biology. I am here to help Joy, Brennan, Paige, and Fonzie describe the reaction between myself and acids and bases. You'll notice that as time passes, my body changes from pink to clear because I'm in an acidic environment. If I were in a basic environment, I would stay pink. I turned clear because the acid diffused through my phenolphthalein gel bod.
We turned clear quite quickly as opposed to the other gel shapes from other tables because we were thin and medium-sized, so the acid could diffuse quickly through us. The other tables had thicker cutouts, which made it harder for the acid to diffuse.
Diffusion is the movement of molecules from an area of greater concentration to an area of lesser concentration. Speaking of diffusion, there is another type of diffusion in the potato lab. It's called osmosis. Osmosis is the passage of water from a region of high water concentration through a semi-permeable membrane to an area of lower concentration. In this lab, they will use potatoes in different concentrations of sucrose to model diffusion.
The purpose of this lab is to identify the concentrations of the mystery sucrose solutions and use the solutions to determine the water potential of the potatoes. The materials used were potatoes cut into 4 mm rods, cork borers, 5 250 milliliter beakers, solutions of 1 molar, 0.8 molar, 0.6 molar, 0.4 molar, and 0.2 molar sucrose, a petri dish, a scale, a scalpel, tweezers, and a ruler.
Hey, it's Tina here. Now let's talk variables. The independent variable was the solution, the dependent variable was the mass, the constants were the potatoes and the amount of beaker solution, and the controls was the beaker and potato sizes. Their hypothesis was that if they put the potato slices in beakers with sugar, the beaker with the most sugar will have the smallest potato, and the beaker with the least amount of sugar will produce the biggest potato.
First, they labeled beakers A through E. Second, they filled each with a solution with a different amount of sucrose concentration. After that, they bore 5 potato rods. Next, they measured the length of the rods and their weight. After that, they dropped the potato rods into the solutions at the same time. They left the solution overnight. The next day, they took the rods out of the solutions and measured their length and weight. The data is as follows. In beaker A, the potato's original mass was 0.9 grams. The next day, its mass was 0.7 grams. Its original length was 4 millimeters, but the next day, its length was 3.7 millimeters. In beaker B, the original mass was 0.9 grams, and the next day, it had a mass of 1.1 grams. Its original length was 4 millimeters, and the next day, its length was 4.3 millimeters. In beaker C, the original mass was 1 gram. Its next day mass was 0.6 grams. Its original length was 4 millimeters, and its next day length was 3.7 millimeters. In beaker D, its original mass was 0.9 grams, and the next day its mass was 0.7 grams. The original length was 4 millimeters, and the next day the length was 3.6 millimeters. And finally, in beaker E, the original mass was 1 gram, and the next day the mass was also 1 gram, and the original length was 4 millimeters, and the next day its length was 3.9 millimeters.
They observed that A, C, and D beakers' potato slices were floating, which meant that the potato was less dense than the water. After 12 minutes, B's potato sank to the bottom.
Because of this, the group guessed that C was the 1 molar solution, D was the 0.8 molar solution, A was the 0.6 molar solution, E was the 0.4 molar solution, and B was the 0.2 molar solution. This order was correct, and later the water potential was calculated, which further proved that this was the correct order because the water potential for the 1 molar solution was negative 24.5976, negative 19.67808 for the 0.8 molar solution, negative 14.75856 for the 0.6 molar solution, negative 9.83904 for the 0.4 molar solution, and negative 4.91952 for the 0.2 molar solution. Water potential is the free energy per mole of water, or the measure of the relative tendency of water to move from one area to another. The more negative the water potential is, the more solute is in the solution and the less likely water molecules are going to move. If there was a high concentration of solute outside the cell as opposed to in the potato, then we had a hypertonic solution and the potato would lose water and shrink. This was how the group knew that C was the solution with a concentration of 1 molar because the potato it produced was the smallest. Solution A lost 22.2% of its mass and 7.5% of its length. Solution B gained 22.2% more mass and gained 7.5% more length. Solution C lost 40% of its mass and 7.5% of its length. Solution D lost 22.2% of its mass and 10% of its length. Finally, solution E didn't lose any of its mass and lost 2.5% of its length.
The solution with the water potential closest to a plant cell is the solution with a concentration of 1 mole. This is because plant cells usually have a low water potential, which means it would have to have a large number. The water potential for the 1 mole solution was negative 24.5976, which would mean that the solution had a really low water potential, similar to a plant.
This experiment could be done in an animal cell, but only in hypertonic solutions. This is because if an animal cell were to be placed in a hypotonic solution, it would burst and there would be nothing to measure. However, this experiment could be done with a fungal cell because fungal cells have cell walls just like plant cells, and the cell wall would keep the cell from bursting. If we did this experiment again but left the potatoes out to dry before putting them in the solutions, the amount of water inside the potato would be lower than the amount outside the potato. This would cause the potato to take in more water, and the solutions with less sugar would have bigger potatoes. The solutions with more sugar would have smaller potatoes. Some possible errors for this experiment could have been that the potatoes weren't all the same weight and did not have the same amount of water in them. Also, the group did not measure the diameter of the rods, and the group may not have dropped the potatoes in at the exact same time. And when taking the potatoes out of the solution, they could have squeezed out some of the water that the potato had soaked up.
We hope you enjoyed our water potential, osmosis, and diffusion adventures.